We became interested in working on XMRV because when we started, we thought this could be a pathogenic virus that's causing prostate cancer, chronic fatigue syndrome, and perhaps other diseases in humans. And we really wanted to determine whether that's true, and if that's true, can we find ways to treat people that have this virus. We've done studies to look for the virus's ability to replicate in people, and we find that it doesn't. There's a cell line that's derived from a prostate tumor called 22RV1 that makes XMRV virus that is essentially identical to all of the viruses that have been reported. So the assumption was that this cell line was derived from a prostate tumor from a person who had been infected with XMRV. We were looking to see how this virus got into this cell line. So we wanted to look at some of the earlier cells as close to the beginning as we could get. We were working with these tumors from the immunodeficient mice, and we found a virus that looked kind of like XMRV, and we worked hard to clone the whole virus, so we had the entire sequence of the virus. And while this was going on, we were collaborating with John Coffin, who has a lab at Tufts University. He was chasing down another virus that looked like XMRV. But we didn't know the relationship between these two viruses. In about mid-December, we had just completed the sequence of our virus. John and his student Oya had gotten almost all of the sequence of their virus. And John kind of looked at our virus and said, you know, this virus looks a lot like the opposite of our virus. So let me take a closer look. So I sent him the sequence. My wife and I were going to go to Egypt for a 2-week vacation that evening. I was late, so I went home. By the time I got home, John had emailed me already and had compared the two sequences and had said, oh, these viruses are exact complements of each other with just a few crossover events. They will generate XMRV, which is nearly identical to the XMRVs that we know so far. So I got this on my iPhone, and I was so excited. I kept thinking, oh, this is so cool, this is so cool. But I was running around packing, and we got in the car. I didn't have a chance to email him back. So we rushed to the airport. We were late getting to the airport. We got into the plane, got our seats, and I took my iPhone out, and I emailed him and said, that's awesome. And that's when they said, turn off your electronic devices. And then I was out of email contact for almost 2 weeks. But it was a very exciting moment because generally scientific discoveries are kind of a gradual process. You get a clue, you get another clue, and you kind of, it happens sometimes over weeks or months. And this was not like that. This was just immediate, bam, this is the answer. And we knew it right away. So it was one of those eureka moments. We found that the virus actually was born in the mid-90s in a laboratory experiment, which pretty much tells us that the virus cannot be causing any of these diseases. So the obvious conclusion is that it must have spread through laboratory contamination.
I think we're seeing now that tools that we use in the lab, reagents, for everyday research are contaminated with mouse DNA. These are things we never knew at the time. The other thing is that as scientists, we share reagents. We share reagents that we maybe didn't know were contaminated. And that's how things can pass from lab to lab. That's what we do as scientists. We share reagents. We hope we don't share problems, but it may be in this case that XMRV is circulated with certain cell lines that it had infected and so on.
It's certainly a cautionary tale that people, as new pathogenic agents are discovered and thought to be associated with diseases, people really have to be diligent to make sure that it's not due to laboratory contamination. Above all, the results need to be independently confirmed by multiple investigators before people can draw the conclusion that, yes, this particular virus or bacteria is causing or is even closely related, associated with a disease, let alone the cause of the disease.
There has simply been a mounting body of evidence that suggests this was contamination. But the work of Drs. Patak and Coffin, this for me as a scientist was the icing on the cake, to see exactly where this virus came from, to be able to trace its history almost 20 years, and to be able to show, I believe conclusively, that XMRV is a recombinant virus, and it's never been in patients and it's never been isolated from patients.